Glucose cannot be used directly by the cells as energy, but it can be used to generate the energy source, ATP. How do cells make ATP from glucose? To understand this, we need to describe the main stages of glycolysis, explain what happens during the Krebs cycle, explain how ATP is synthesized during oxidative phosphorylation. Cells form ATP from the breakdown of glucose in a process called cellular respiration. There are two types of cellular respiration: aerobic respiration and anaerobic respiration. Aerobic respiration is much more efficient and produces much more ATP than anaerobic respiration, which only takes place in the absence of oxygen. The first step of both aerobic and anaerobic respiration is glycolysis. It takes place in the cytoplasm. First, two molecules of ATP are hydrolyzed, adding two phosphate molecules to a glucose molecule to form hexose bisphosphate. Hexose bisphosphate is more reactive than glucose and splits into two molecules of triose phosphate, which is phosphorylated again. Hydrogen is transferred to NAD to form reduced NAD. The molecules are converted into pyruvate in enzyme-controlled reactions, generating two molecules of ATP each. 4 molecules of ATP are produced, but 2 are used, meaning there is a net increase of 2 ATPs. The rest of the process of aerobic respiration takes place in the mitochondria. Pyruvate is moved into the mitochondrial matrix by active transport. The first step of aerobic respiration is oxidative decarboxylation of the pyruvate molecules. This is sometimes called the link reaction. Carbon dioxide and hydrogen are removed from pyruvate to form a 2-carbon acetyl group. This group binds to coenzyme A to form acetyl coenzyme A. The hydrogen is transferred to NAD to produce reduced NAD. The acetyl coenzyme A moves to the next stage of aerobic respiration, and the carbon dioxide diffuses away to be removed as waste or used in photosynthesis. The next stage of aerobic respiration is called the Krebs cycle. The acetyl group is delivered to the Krebs cycle by acetyl coenzyme A. It combines with 4-carbon oxaloacetate to form 6-carbon citrate. Citrate undergoes decarboxylation and dehydrogenation to produce 1 reduced NAD, carbon dioxide, and a 5-carbon compound. Further decarboxylation and dehydrogenation reactions take place to produce oxaloacetate, which will start the cycle again. 2 carbon dioxides, 2 more reduced NADs, and 1 reduced FAD are produced during each cycle. 1 molecule of ATP is produced by substrate-level phosphorylation. Most ATP generated by aerobic respiration comes from oxidative phosphorylation, as electrons are transferred along an electron transfer chain. The hydrogen atoms collected by the coenzymes NAD and FAD are delivered to electron transport chains in the cristae of the mitochondria. The hydrogen atoms dissociate into hydrogen ions and electrons. Electrons flow along the electron transport chain, releasing energy as they reduce and oxidize electron carriers. This energy is used to create a proton gradient, causing protons to diffuse through ATP synthase and generating ATP. At the end of the electron transport chain, the electrons combine with hydrogen ions and oxygen to form water. Oxygen is the final electron acceptor. Cells use ATP as their energy source. ATP is generated from glucose during cellular respiration. In the cytoplasm, glucose is split into pyruvate during glycolysis. The process then moves into the mitochondria, where Krebs cycle takes place. Coenzymes, reduced during the Krebs cycle, deliver hydrogen atoms to the cristae, where electrons are transported along the electron transport chain, generating ATP by oxidative phosphorylation.